good morning, everyone. Want to thank the organizers first for organizing this interesting conference. Okay, so I'm the head of the lab of computational systems and synthetic biology at Tel Aviv University. And in my lab, we engineer cells and viruses. Among other, we actually try to affect the fitness of the organisms or the viruses. And I think that my research is very relevant to this conference, and we try to convince you that this is the case. And the major project that I will talk about is ongoing project where we try to encode information, orthogonal information. It can be information not related to biology, okay? It can be, for example, a picture of you, okay? On the genome of living organisms. But before I will describe this project, I will start with a general introduction, a relatively long introduction that will help you understand why I think that this project is related to this conference about the origin of life, and also a lot of background related to the understanding of the project itself. Okay, so we start with general introduction and motivation. Okay, so in my lab, we study a process which is called gene expression. I guess some of you know this process. This is the major process in biology, in life. And this is actually the process by which information encoded in the genetic material, in the DNA or RNA, is used eventually for generating protein. Not only the sequence of amino acid, but the rate of generation of the protein. This is also encoded in the genetic material. Thus, actually, this is, I think gene expression is the essence of life. This is life. This is a process that actually transforms information in animated molecule, okay, a dead molecule, the DNA, to a living cell with various proteins that interact with the environment. Okay, and this process includes many steps. Usually, people know the major steps, which are transcription of DNA to mRNA and the translation of the mRNA to protein by the ribosomes. But there are many additional steps, many processing steps of the RNA, stability of mRNA, stability of proteins, many additional steps and sub-steps. Don't have time to talk about all of them. If there are questions, please tell me, okay? Because I see that someone writing in the chat something, and I'm not sure if you try to ask something. You can ask questions during the talk if you are able to do it. Okay, so one question is where this information related to gene expression is encoded. Okay, where in the genome it is encoded. Okay, so this is important information. So the traditional model suggests that actually we have a modular situation. Okay, for each protein is encoded by a gene. The sequence of amino acid of each protein is encoded by a gene, the set of the sequence of amino acid. And upstream of the gene, we have a region called promoter, where the information related to the dynamic of generation of the protein, okay, or the rate of generating the protein, or the gene expression, is encoded. Okay, so this is a traditional model, very nice model from the engineering point of view, but it is wrong. Okay, so today we understand that actually information which related to gene expression is encoded all over the genome and specifically inside the gene itself. Okay, so if you look at the gene in even in the coding region, where we have the genetic code that code the sequence of the amino acids, we have many overlapping codes in the same region that actually encode the different step of gene expression. Okay, so there can be in the same region codes for transcription, translation, splicing, all aspects of gene expression, the stability of the mRNA, and the genetic code. Okay, so if you change a nucleotide in this region, you can change both the amino acid content, but also at the same time you can change that different aspects of gene expression. And another point that I want to emphasize the fact is the definition of silent mutations. Okay, so let's talk about for a minute about the genetic code. Okay, so the genetic code, I guess you should know it. Okay, this is a very important code. that translate. It's almost an universal code that appears in almost all organisms and viruses today that actually translate triplet of nucleotides to amino acids. Nucleotides are the alphabet of the genetic material, which is DNA and RNA, and amino acids are the alphabet of the proteins, okay, the building block of proteins. And this is the genetic code. You can see the table here. I guess most of you know this. And you see triplets of nucleotides are translated. This is according to the code to different amino acids. And you can see that the code is redundant. Actually, we have more triplets, and we call these triplets codons. There are more codons than amino acids, around 3 codons per amino acid. Okay, this is the redundancy. And usually, for a long period, people call the mutation, actually also today, people call a mutation that change a codon to a synonymous codon to another codon of the same amino acid. They call it silent mutation. Why? Because they assume that the important actually code is the genetic code, the sequence of the amino acid. And if we change a codon to a codon that do not change the amino acid, actually we do not affect the fitness of the organism. And this is, of course, wrong. So today we understand this is not the case. As I mentioned in the previous slide, actually we can change gene expression code when we do it, and this can affect the fitness. Okay, this is, I think, relevant to all organisms that I know, also to ancestral organisms, and probably version of it relevant to pre-cellular evolution on Earth. Okay, so in my lab we try to we develop models for deciphering different gene expression code. We introduce these models into quantitative, rules into quantitative models. And this can be a simulation of gene expression or different predictors. And we use these predictors for engineering cells and viruses to solve various biotechnological problems. So this is generally what we do. But also we are interested in the basic science. And this type of conference many times inspired me, because by learning how life evolves, I can think about how to generate new life or new cells or new viruses. It gives me many ideas, and I think also what we are doing, I hope you will be convinced later, can help you think about life, also the origin of life and the evolution on Earth. Okay, so I will give you now a few slides of application, what we are doing, and then we will start with the description of the project itself. So what we can do with these models, so for example, we have many projects that we engineer viruses. We can do silent mutations into viruses. We change the gene expression code, but not the encoded protein. So we can attenuate the viruses and get vaccines, okay? So you can see in this graph, for example, take a certain virus, Zika virus. This is the wild type. This is the replication rate of the titer of the wild type. This is the titer application rate of attenuated viruses. So you see some of them are very, very attenuated. And we perform mice experiments showing that when we vaccinate relevant mice with this attenuated virus, we do not kill them, but eventually they are vaccinated against wild type Zika. They kill most of them, but not the vaccinated mice, okay? We can also engineer cells and treat them as factories for generating different proteins, okay? This can be therapeutic protein, food industry protein, or energy. And the idea is to, again, optimize gene expression, making the cell a better factory, generating more proteins. By understanding how the cell will evolve, I can further improve them for my objective. And we also showed that we can actually take cells, for example, yeast, as cerevisiae, introduce site mutations to the cell and also improve the growth rate of these cells. Okay, so this is very interesting because it suggests that I think growth rate is something that undergo extensive evolution, both in cellular organisms and precellular life on Earth, or the origin of life. And actually, we show that we can further improve this process. The evolution is not optimal in this, objective, which I think one of the major objectives of evolution, at least of microorganisms and viruses. And we can also engineer cells, for example, E. coli and viruses, for example, to actually become biosensors. This is an example. We generate an E. coli that can detect explosives, okay, by engineering introducing various new genes into the bacteria and engineering their expression levels by our models. Okay, so this was an introduction. And now when we talk about our project, I think it's very interesting project and relevant to this conference. So the idea here, we want to actually, as I mentioned at the beginning, you want to take a certain organism, for example, a bacteria or virus, and write a new information on the genome of this bacteria without affecting its features. Otherwise, the information will be deleted. This is a very general idea. Why we want to do it? Maybe it may sound a little bit crazy. So there is, I think, two objectives. One of them is basic science. We want to learn what is the efficiency of the genetic materials in terms of how much it is populated with codes. Okay, how much information we can add to this genome of living organisms. I think it can help us understand also the origin of life and the evolution of these genomes. And so this is one aspect. We want to, another aspect is the fact that we try actually to model all the evolutionary process as a communication channel. And I think this is an approach that can be used to study evolution in general. But there are also other objectives that are more biotechnological objectives. So for example, as I mentioned, we use today bacteria and viruses as small machines in the cells that can, for example, be biosensor or solve different problems, therapeutic problems, for example, fight cancer. And I cannot think about engineering a machine, even a small engineering machine, that we cannot write or read information, that do not write and read information during its process, okay? As an engineer, this is my knowledge. Okay, so what is the challenge? The challenge is to actually write information into organisms that undergo evolution, okay? We want to write information on the genome of living organisms. And during evolution, as you know, there are different types of mutations that actually can delete the information that we wrote. And actually, also, the information that we write can affect the fitness of the organism, the growth. So if we change a mutation, we introduce mutation, write something on the genome of the organism, and actually affect its growth rate, it may disappear. Okay, if this is a microorganism, the growth rate will decrease for a few percent, and it may disappear. It cannot compete with the wild type bacteria. Okay, so these are and there are other challenges related to the technology. So we assume today that we can write and read information only to a certain short region in the genome, a certain gene. We want to write it inside the gene, because if we do not write information in a gene, but put it in a genome in a different location, not in an essential gene, this information can be deleted, okay, by the organism during the evolution, because microorganism knows very well how to delete, okay, during the evolution, delete part of the DNA that do not affect the fitness, okay? So we try to interleave the information inside the essential genes so that the organism cannot delete it, okay? In this work, it's very, very multidisciplinary. We use here tools from coding theory, information theory, synthetic biology, molecular evolution, and biophysics. So this is the general idea, just again to give you, I think it's a little bit strange idea, so I try to make it simple. So we have a source of information, for example, a picture of you, okay? We can describe every type of information as a sequence of bits, okay? We actually will translate this bit to nucleotide. I will talk about it later. And we want to write this information in a certain gene, which is essential gene of a certain microorganism, such that the microorganism will not be able to delete this information, and it will not affect its fitness too significantly, so this organism will disappear. Okay, we want to be able to reconstruct the information. And due to limitation, technological limitation, actually we write this information not on one bacteria, but many strains of bacteria. In each of them, we write part of the information in the same location, in the same gene, and the concatenation of all this information is the written message. Okay, so after we write this, we have a community of bacteria. They undergo certain evolution. Maybe we write a year, maybe 10 years, maybe a million of years, I don't know. And then we want to sequence the genome in this region and read the information. Okay, this is the idea. We read the nucleotide composition and we translate this to bits. Okay, so this is the general idea. And so the project includes the following steps. We start with modeling the local problem as a mathematical problem, communication channel. You need to estimate the parameters of the model, the probability of, for example, of mutation, of the effect on the fitness. We need to estimate it. Otherwise, this is not, practical. We analyze all this model mathematically, try to develop a code, and also understand what is the capacity of a genome of a living organism to introduce additional information that we reliably can read. And then we perform some simulation and experiment, which is ongoing experiment, to see how well is our approach. Okay, so this is the idea. So what happened in our communication channel? Okay, what can happen with the information that we write and read into the bacteria? Okay, what the evolution can do? Okay, this is actually part of our model. So the first point is the fact that, again, if I introduce certain mutation, we can affect the fitness of the organism with the information written on it, and the organism can disappear, okay, because the growth rate will decrease, and then large part of the message will disappear. Okay, this is one possibility. Another possibility, another point is the fact that actually we cut the message to short messages, as I mentioned, and there can be permutation of the message that we need to add some kind of identifier to each of the messages to be able to concatenate the messages at the end. And the last thing that can happen is mutations, point mutations. Okay, there can be different mutation in the message that actually will introduce errors. Okay, so this is our channel. So eventually we write a set of sequences, as you can see here, and eventually after we write it into a set of bacteria. Eventually, then we get this situation. There can be mutations. Some of the subsequences can be deleted, and we want to generate a code that can reconstruct the original information. So this is a very general idea what we are doing. Of course, I do not get into the technical details because we need a background on this. It's related to information theory, encoding theory, but I want to talk about the big picture. Okay, so these are the different steps of encoding the information. Okay, so we have a source of information, a sequence of bits. Okay, this is the transmitter, and the bits again, they can be your picture. Okay, something orthogonal. So the first step, we add additional bits to the information to be able to protect from errors. Okay. And there are many ways to do it. This is usually done in communication. For example, suppose you want to transmit a bit 1, and there can be an error that can a small probability that it becomes 0. Okay, so the very trivial code you could think about, add at least instead of sending 1, send 1 3 times. Okay, add 2 bits, additional bits with 1 1, and then the probability that two of the ones will become 0 is exponentially lower, and this allows you to protect from errors, better protect from errors. Okay, so we add additional bits to protect from errors. Now we need to split the message, as I mentioned, and we add identifiers to the different sub-messages. And then we actually use a modulator that actually translates bits to the DNA nucleotides. And we are doing this, among other, by considering the different gene expression code that I mentioned. So when we actually write something on the DNA, we actually use all our models that consider the possible effect on the gene expression, and we don't want to affect gene expression. And of course, we don't want also to affect the genetic code, the encoded protein, because we don't want to affect the fitness of the organism. Otherwise, this message will be deleted because the growth rate of the bacteria will be lower. Bacteria or the microorganism will be lower. Okay, so this is the general idea. So we introduce a way to write this information, be able to understand what are the different bits, but without affecting the fitness. Okay, so this is what happened in the transmitter, what happened in the receiver. So after long period, maybe 10 years or million of years, we want to read the information. Okay, so we have this data, which is DNA sequences with many errors, and some of them are missing. So the first, we use the demodulator to translate this to bits, okay? And then we get a set of messages in binary code. And then we actually use the identifier to generate the long message that you try to submit. Use the error correcting code to fix errors, and we get a message. Okay, this is the general idea. So we developed a mathematical model that describes all these
Tamir,
aspects.
you have 10 minutes from now.
Okay, no problem. Okay, so we developed a mathematical model that describes all these aspects that I mentioned. The legal dictionary, actually the mutations that are legal and will not affect the fitness of the organism, the probability of a message which is deleted due to the effect of the fitness, the permutation of the different sub-messages, and the substitution probability. Okay, this is a mathematical model that describes a channel. A channel, this is a mathematical entity that is used many times in the coding theory, people that actually develop codes, okay? So actually we suggested to look at evolution of living organism as a channel, okay? I think this point of view very interesting and maybe can be used for understanding evolution and also precellular evolution, okay? So now we need to estimate the parameters of the model, okay? All the parameters that I mentioned, for example, the substitution probability, the deletion probability, the effect of mutation of fitness. How can we estimate them if we do not know how to estimate them? Actually, all of this is very, very theoretical idea, okay? But we need to estimate them, and the way to do it is to use genomes of organisms that live today. For example, take organisms from the tree of life, as you can see in this picture. You can take organisms from all the domains on the tree of life. This is Luca. These are the three domains, bacteria, and eukaryote. And actually, when we take many orthologs, okay, version of the same gene from many organisms, we can track what can happen in evolution, okay? And from this, we can estimate, at least get an initial estimation of the different parameters of the channel. And from this, we can estimate both the performances of the channel, and also we can think about a way to optimize our codes, okay? So that's a general intuition how we can do it. So this, as you can see here, this is an example of an alignment, okay, of a certain gene that we want to write information into the gene, okay? So each row is one organism, each row describes a version of the gene in different organisms, okay? So if you look at a certain position, you can see that, for example, this position, it is very, very conserved. But it means that if I will change this position, try to write something new, probably it will affect the fitness of the organism, because this is actually important functional position. Okay, this is what we suspect. And another example, another extreme is this position where there are many substitutions. Okay, so in this case, if I introduce mutation, probably it will be substituted, and there will be an error. Okay, I will not see the original mutation. Okay, so we need eventually we need to find positions that are not something in the middle, not very conserved, but not extremely non-conserved. Okay, so this was the general intuition. So based on the mathematical model, we define what is called capacity. Okay, capacity of a communication channel is the maximal rate of information that we can transmit and eventually read it reliably without errors. Okay, so we can mathematically estimate the capacity of the channel, the mathematical channel that I described, and we use a different theorem to prove it. This is not important here. The bottom line that
sorry to interrupt. You have 5 minutes from now, including question and answers.
Oh, in 2 minutes I will finish. 1 minute. Okay. So eventually we found that our codes are 85% of the capacity, so we are very close to optimal. And I think this is very interesting, suggesting that we have a nice code. And what we are doing now, we want to actually experimentally check the code. Okay, so what we did, we actually analyzed. We are working with cerevisiae. We analyzed all the genome of all the genes of cerevisiae and found that in some of them we can write information that we can start with in the efficiency of which is 90% of the capacity. And now we are in the process of performing an experiment where we write this information and you want to read it after 1 month, 2 months, 3 months, etc. Okay, to actually learn how well is our code. Just to give you some intuition, I think this will be very interesting for you. So we decided to write 66 English words. These are the beginning of our paper, okay? And to do it reliably, we actually need 23,000 nucleotides on total. Inside actually we want to write it inside an essential gene, okay? And this is why it's so hard to do it, because we don't want to actually change the fitness of the organism. Okay, so the bottom line is that it's very hard to write. We need many nucleotides to write this information reliably into essential genes. So to summarize, I try to cover many interesting stuff, I think. So first, I talked about the fact that in the genome we have many non-modular overlapping codes in addition to the genetic code. I think this is very relevant to this conference. Many of these codes are related to gene expression control. So that's a new approach to look at evolution, which is based on coding theory and describing the evolution as a channel, a communication channel. I introduced a new approach to introduce that can add additional information to the DNA and estimated how well we can introduce this type of information. And I think this, the result that I showed you is very interesting and raises many questions. For example, what is the information, the ability to introduce information to ancestral organisms versus organisms that appear today, and what is the evolution in terms of information capacity of living organisms? Okay, so the ancestral organisms of pre-cellular organisms were less efficient in terms of the evolution of the information that were encoded in this their genome. So I want to finish with thanks. So Professor Yitzhak Tamir is a collaborator of this study, and these are my students. I want to thank Alon Akiva specifically. He was the first author, but many other students also helped. And thank you. I'll be happy to answer questions.